This is a production of Cornell University.
Thanks, Larry. Good to see this huge crowd here today. I'd like to start out by introducing the people in my group that have actually been working with hemp diseases. I'll tell you what I think about hemp diseases, but I'd like to, first of all, Jamie Cummings is here, who has been with my program for a number of years. It's now with the IPM program, but kind of got us started on some of our hemp work. Kevin, you want to raise your hand there? Kevin Myers, and now Jen Starr is my research and extension support specialist. So what I'd really like you to know is when you're out in your hemp fields and you're finding things you think may be diseases, if you notice something, say something, let us know, get in contact with one of us, and we'd like to diagnose the problem. Like any other new crop, there are a set of pests, insects, weeds, diseases that will be eventually a problem in the production of that crop. And of course, this is the second year out with this new crop in New York State, unless you go way back when it might have been grown previously. But in recent history, it's all new to us. And that includes the plant pathologists that are working on this. So with our 1 year of experience last year under our belt, we saw about a dozen different microbial organisms that we found that cause disease on hemp. And these vary everything from things that cause a rotting in the root system that can affect the seedling establishment of the crop, to things that cause a lot of fungi that cause spots on leaves, blighting on leaves. And then we saw things also in the buds, in the heads, the flower parts, the seeds. Very often we found something we would call gray mold, botrytis, a fungal problem infesting the buds. But by and large, the message is that most of the industrial hemp that we've seen has been pretty darn healthy. As you may know, and you're going to hear more about today, there are different classes and uses of industrial hemp, from the CBD types to the grain types to the fiber types, etc. And you're going to hear about that. And so there's a lot of different genetics out there in these fields, and I'm sure Larry and others are going to talk about that. But that means there's a lot of diversity in the crop that brings about a corresponding diversity in the microbes that cause disease. So we've definitely seen in some of the CBD types more of a susceptibility to some of the leaf spot pathogens, for instance. So I'm not going to try to clutter your minds with a lot of Latin names of microbes today, but again, what I'd like to leave you knowing is that we're trying to get an early handle on these things. Eventually, what we'd like to see is working with Larry is to try to make selection in these different classes where he's doing the breeding and seeing if we can't increase the level of natural genetic disease resistance, which would be our first line of defense. I think you're all aware, and you will hear from others here today, that because of its status as a non-crop officially at this point, there are none of the FIFRA labeled materials for control of pests, and that includes diseases, even things like seed treatments that Alan's going to talk to you a little bit more about. But eventually, we hope as the classification of this crop moves along from the federal government to kind of keep up with what's going on at the state level, that we will be starting to look at different kinds of pest management materials, including synthetic chemicals, as well as organically approved materials for organic production. Right now, the best thing we can do is start out with some sound cultural practices. And I think you'll hear this over and over again today. Rotation is our greatest friend. There's less likely to be disease impact if you grow in sequence fairly unrelated crops. So hemp being a broadleaf crop, probably its best rotational crop partners would be things like small grain cereals and corn, the grasses, the monocots. We have some commonality in some of the pests, especially across some other broadleaf crops. We're going to learn more about that as we go, but one of the biggest problems we have is white mold, that anybody that grows soybeans or vegetables is well acquainted with. And so a rotation with soybeans or some of the more susceptible vegetable crops is probably not the greatest choice because of susceptibility to white mold. And we're learning other connections like that. A lot of the disease organisms, some of the fungal leaf spots, are very specific to a particular species of plant. And so it's not so much a problem there, the rotational sequence of crops, but looking at variation in the varieties, in the cultivars of industrial hemp. Some are going to be more susceptible than others. So as we go along and working with Larry and his program, I think we're going to learn more and more about that. So again, my message to you here today is if you see something that appears that might be disease, we're in the business of diagnosing that and trying to get a handle on it, and would love to work with you. So, thank you.
Thank you. I think we have a couple of minutes for questions. Sure. Are there any questions for Gary?
How do we get those to you or show you them, or how do we know about the people?
Okay, so we have a laboratory in Ithaca, and I don't know, you've got, maybe we can put that on your on the Hemp website too. That might be the easiest thing as we do, but for diagnosis of plant diseases, we can put that on the website. Otherwise, my email is gcb3@cornell.edu, but I think that's probably the best thing we could do is put something on the general web, Cornell Hemp website. I'd be glad to give you a card too today. Yes, sir. You know, the question here is, do we have a listing of varieties and by diseases about relative susceptibilities? And quite honestly, we're so early into that cycle of trying to understand that we kind of have a shopping list of things we have seen. But to partition it out that finely, we're not there yet. And of course, this is a very dry, warm, early part of the growing season. We'll see how that ends up. But up to this point in the growing season, there has not been an intensity of diseases. We've had to look very hard to find any at all. So good news, bad news. But last year, we cataloged quite a few different pathogens. As far as which ones are going to become really severe, I mean, that's still a lot of guesswork. And there's not even really an extensive world literature on this, interestingly enough. So we're very much in the learning mode here. Existing as in biologicals or? So question's a good one about if we used any kind of inhibitors, natural products, whatever, the soil biome itself. The short answer is no. I think there's a lot of possibilities there, but with the learning curve, we're here somewhere. But those are some of our aspirations as we move along with this. And I might point out, as I'm sure Larry will, that we're not in this by ourselves. There are 35 other states. Yeah, so the question was referring to connecting with the State Department of Environmental Conservation, which we are in good communication with the DEC, but really, even above that, there are no EPA permits. There are no national permits for use of any of these plant disease control materials. So even before it got to state regulatory, there's nothing allowed. Now, there are some things that are on the grass list that are used for food processing and whatnot. And maybe you're going to deal with that a little bit. Maybe Alan's going to talk a little bit about that. But there are some materials that might be useful in certain things, but in general, we don't really have any tools in our arsenal right now for disease or insect control. Yes, sir. Yeah.
Yeah. The DEC, I think it's great. Talk to the lady in charge of chemicals. I submitted a list to her her, and she was, within a week, got back to me on my list of yes and no that I could do.
Okay, maybe we can talk about that a little later, but thank you. Thank you.
Video, so I will be here. First of all, I want to make sure I got in a little bit late. If any of you came in late, this is the handout that I'm going to be going through. There's extra copies up here if anybody needs those. Possibly you need to grab some. Hey, just raise your hand if you'd still like to have a copy of this. Maybe some people in the back. Okay. So I'm based here at the Bergstrom station, AgriTech, Cornell AgriTech, and I'm getting used to saying that right now as we've been at the New York State Agricultural Station for the past 130 or so years. But my area is in seed science and technology, so I'm kind of, by nature of my program, but also my own interest is kind of to work with new crops, especially those that can have really a benefit for New York State. And I think that the hemp is really one that fits into that, where there's many seed challenges. It is really a new crop. As Gary talked about in his presentation, there's some things we kind of expect. There's some things that we simply haven't ever worked on. You can't look up somebody else's work to really know that much. So we're really trying to establish some really sound science. That science, that applied science, can be translated into outreach and extension to benefit our New York agricultural community. If we kind of, how did we get here? If we look at the 2014 Farm Bill, it allowed states to go ahead and to work on research on hemp. Several states have done this. New York, we really got into this in a big way last year and even bigger way this year. I contacted Ag and Markets, and we're looking at 3,200 acres of hemp that are planned to be are grown here in New York State. This side of the Mississippi, some of the major states, Kentucky, North Carolina, and then you go to the mountain states of Colorado, the largest, in Oregon and the West Coast. So quite a substantial industry. And we also learned a lot from other countries, in particular our neighbors to the north. Canada has over 110,000 acres, so more of a mature industry there that we can rely on for seed companies, for getting plant materials as far as seed sourcing. So in your picture, probably if you've already been growing hemp, you know what the seeds look like. If you actually put a seed on top of the photo, this is about the actual size of the seed. This is just one of the commercial lots that was used extensively last year in some of the farmer trials called ANCA. And we did not adulterate the photo there on the left. There is just a huge range of seed sizes. And if you're working with any other crops, if you work with field crops or vegetable crops, you know the seed companies have done a much better job of sorting seeds by size. So that's kind of the first thing is in we deal about irregular shaped seeds. We've done enough work with those small seeds that really just produce runt plants. They're really kind of just there, part of the seed count, but they're really not that productive. Anyway, starting off with those raw seeds, my goal, again, working with Gary's program, is we're trying to be proactive and looking at seed treatments for the future. In the 2018 Farm Bill, as we look at that one, if all goes well, hemp should be exempted from the DEA, which is really going to open up a floodgate for having it being grown just as any other vegetable or field crop. So it would also allow pesticides. We're trying to be proactive in that and looking ahead. Those of you working in the vegetable or specialty crops area are familiar with the IR4. IR4 works with university partners and others to foster the registration of materials, both chemical and biological or organic. And so IR4 already has a program. They're allowed to work with hemp. There's a program now at NC State on weed control. And so we're kind of trying to get our foot in the door for next year for 2019. So one part of that is, besides the different materials that we'll be applying, either chemical or biopesticide, is how we're going to apply that. So my laboratory's done a lot in that area. So if we use our standard kind of a slurry technique, that's what the grain is, yeah, we're getting materials on the seed and the color, but you can see a lack of uniformity. So we've tried to spend some time of kind of upgrading the seed technology, and so now we can see with the proper use of different seed coating materials, we can get a very uniform end product, and that's really what we need to as we're going to be moving into that. Again, kind of working to try to establish ourselves with an IR4, and I've worked with that entity for many years of, again, looking at chemical seed treatments for direct seeding, but also to look at biopesticides, which would be your organic material. So that's kind of next year's presentation, so again, I'm just kind of looking ahead for that. We're going to change gears now into the seed testing, seed quality aspect. Standardized testing procedures are in place. They've been in place for many years, as it is for other crops, field crops. The same, using a roll towel test. We're looking at 2 samples of hemp, and these are from testing samples. We had 23 seed lots that are being used for the field trials this year, and my laboratory did both the germination and the purity. We'll talk about the purity in just a moment. But if you can look at that photo, you can see the ones on the left. Very poor germination, lack of uniformity, and the photo turned out pretty well, but there's just a lot of mold growth, things like that. So there can be actually internal soil-borne pathogens, soil-borne diseases within the seeds, and they're just going to kind of proliferate. So again, kind of another need for having effective seed treatment in control there. What we'd like to do is have what looks like the roll towel on the right-hand side with very uniform seedling, high germination potential. Okay? So let's go over to the second page of the handout. And there are already, ASCA, which is the official certification agency for the U.S., already has minimum established standards for what the seed lots would have to have. The first one you always think about is the germination. So the minimum germination has to be 80% or higher. And the other aspects are what we call the purity of the seed. We want the pure seed. So inert material, which is just broken seeds, other trash material, other crop seeds can be present, wheat seeds. Any of those would distract from having pure seed. So, and there are already thresholds established there. So what we're looking at here are those 23 seed lots. I've ranked those from high to low germination, and you can see we're all over the board as far as germination percentage. Those germinations, again, using 80% as the threshold, we only have a little over 50% of the seed lots would even pass certification standards to be planted in New York. Now I have a few other areas over here that are highlighted in red on the purity side. So again, the other crop seeds exceeds the certification standards, other wheat seeds that are present. So if we just took these seed lots as representatives, we only have a little over 40% of the seed lots would even pass. So this industry, which has somewhat been of an underground industry, really needs to raise itself up just to put itself to make certification standards. So that's kind of one of my take-home lessons here, is that the seed quality can be good with hemp. It does not have to be poor seed. And if you have other crop seeds, inert material, wheat seeds, that when you open up that bag of seeds and they're there, that is really something the seed companies can easily clean out with seed conditioning equipment. So we really need to have really improved seed lot to really improve the stand establishment potential for this crop in New York State. So at this time, I'll just open it up if anybody has questions. And I probably should have prefaced this because some of you are CBD people. All these varieties are for fiber or for grain. We've not done the testing aspect on the CBD varieties because of limited seed sources, but I just want to preface that.
Money.
That is another big issue. Yes, sir, you had a question? Yes. I'm sorry.
Yeah, I was going to ask you what the testing document number is for the SOP in New York State.
Oh, okay, so basically, what are our standards that we use? So there is official standards for seed testing adopted by the acronym called the AOSA, the Association of Official Seed Analysts. It's the same type of criteria that our state seed testing laboratory would use in Albany as well as other states. So there are criteria as far as temperature in which the seeds are tested, the duration of the test, things like that. So we're really providing prescribed methods that we're following as far as what this germination and the purity tests are done with regards to sample size. We can talk more about this if you have other questions, but basically they're already established. Yes, Sam.
Somebody's not familiar with large-scale commodity recycling. Do you forecast that most large-scale operations will have some kind of seed coating? And does that also include in the organic realm?
A good question. So Zach was asking about, we're really looking at maybe future for next year about what seed treatment. So as far as run it as what I would call as an industrial crop for fiber or for grain, we may have for next season, as early as next season, again, that's very optimistic, that we have some of these materials that's already used on field crops. So it'd be more of a seed treatment rather than a build. Don't think of like a seed pellet and things like that. We're really just looking at a small amount of the active material. Also, they could be biological in nature, organic seed treatment. I'm just a lucky guy, you know. Expect the helicopter. But with the organic seed treatments, again, both the organic grower and a conventional grower could use those, where the chemical type seed treatments would be just for a conventional type of a production, just the way it is in crop seeds. But again, it's the timing. We got to get the 2018 Farm Bill is going to have to go through where hemp is exempted. That will open things up. So again, what Gary and I are trying to strategize is try to be proactive.
Kind of going on this year. What is feminized seed and how are they doing it? Why isn't it working?
Right. For those of you involved in CBD production, you have an option. It depends on what the seed company is providing as feminized seeds. And what are feminized seeds? Basically, is you want to have the plants that you are producing only to produce flowers. You do not want to have those produce male flowers. Hemp seeds are naturally what we call dioecious, means they have, there's male plants and female plants. What we want for just the flower production, where the CBD production would be the highest, is those that only produce female flowers, so as we had harvested at. So what the feminized seed part of that is a particular method that needs to be done during the seed production to produce those feminized seeds, where you're using different materials, silver materials, things like that, to change the sex expression. So basically, think of it in our own kind of our chromosomes with XX and XY. Hemp is the same type of a plant species, with XX being female, XY being male. So basically, is you need to have XX pollen to produce the seeds, to produce the feminized seeds. So it's a rather specialized, the companies, these seeds are extremely expensive. Those of you working in it could be $1 or so per seed. So it's an expensive type of an operation, but one that you would have that high level of, very, very high level of female plants. The other way is if you do not have that, or the procedure is during seed production, still gives you some males.Ose males are really going to have to be broken out during the seed production time, during the crop production time, so you're not having that source of male pollen present there. Good question, John. Yeah, Larry, you want to jump in here on the genetics aspect of that?
Yeah, we need to move on.
Okay, oh yeah.
So I just want to say that some of the grant funding that we have from New York State is to facilitate certified seed production. Seeds are a big issue because federally, the DEA still considers it illegal to move live seed across state lines. So that is a strong disincentive for companies to invest in seed production when it is federally illegal for them to sell across state lines. Now, the Omnibus Spending Act included language that said that the federal government could not expend any funds to intercept that movement. But the DEA still considers it illegal. So when companies are doing their due diligence, their lawyers tell them that. So it has been a challenge for us to work with seed companies to overcome that barrier. Let me also say that New York State is not necessarily the best environment for production of disease-free seed. So when those restrictions are lifted, we may well see an intensification of seed production in those states that have optimum climate for producing disease-free seeds, mainly Oregon, Washington, Colorado. So that's another disincentive for New York seed companies to produce seed here. All that being said, we had a company committed to producing certified seed here in New York State. That was International Hemp Solutions. Was committed to produce certified seed of Yellow-Pacheski, one of our best cultivars from last year. Unfortunately, it took us maybe more than 8 weeks to get that. Took us 2.5 months to get the seed imported from Poland. The first lot was contaminated and it did not get phytosanitary certificate. The second lot was coming right from the Breeding Institute in Poland. So that seed is now sitting in our freezer here and is ready for next year. So next year we should have production of certified Yellow-Pacheski here in New York State. So the question is, are there companies that are producing seed in New York State? No, there are no companies right now producing seed within New York State. New York State Ag and Markets has adopted the AOSCA seed certification standards. That's the American Organization of Seed Certifying Agencies. So we now have a framework for certifying seed and putting a label on hemp seed. By next year, we will have certified seed production in New York State.
You're not required to use certified seed.
There is not a requirement to use certified seed. So, one more quick question.
Are you guys working at all with the medical marijuana, or is there a company in New York State that has medical marijuana?
So, at Cornell University, we do not work on marijuana. So, no, we are not working with medical marijuana companies at Cornell. I'm going to pass it off to Tim Sweeney. He can answer some of your questions and give an overview of the current status of the pilot program in New York.
My name is Tim Sweeney. I'm a policy analyst at AgriMarkets, and I have the privilege of overseeing much of the industrial hemp program. How many, just out of curiosity, how many here are not currently involved in the program but hope to apply? Okay, so I don't think I need to run through the entire process. If you all want to come up and talk to me afterward, I can give you the broader view, but I'll just do a quick synopsis right now. As was mentioned previously, we have about 3,200 acres that could be planted in New York State, approved for planting. If everyone that is approved planted to the extent that they apply for, we have about 3,200 acres. We have 136, I believe, growers, research partners registered with us, authorized by us, and 28 processors. We continue to take applications on a rolling basis for people that are interested in growing for food and fiber. We are not, however, able to take any applications with regard to CBD. Because this is a research project, we We had a lot of CBD applications come in the past, in the first few years, and this year particularly. So we have stopped taking CBD applications to allow us to get a handle on the CBD world and to better establish guidelines for regulating CBD. We are, as I said, we are still taking applications for growers. However, given the lateness of the season, it would probably be advisable if you wanted to apply for the permit to wait until later in the year and apply, because it is a 3-year permit, so you wouldn't want to waste the first year of it and not be able to get anything to the ground this year. With regard to getting seed, as many of you might know, if you want to get seed from an international source, you have to come through us. We hold the DEA import permit. You would submit your request through us. You make all the arrangements with your seed company. You submit a request to us. We then file the DEA paperwork. They send us the import permits. We then give them to you. You give them to the seed company. And your seed has to come to Albany. We have to take possession of it. You have to come and pick it up in Albany. We tried to get around this. We asked the DEA to allow us to meet shipments at farms, and we got a big no. So that just is the way it is for now. As also was mentioned earlier, there is pending federal legislation, federal farm bill. If the farm bill passes, and this is a big if, with the language that's included in the Senate version, this program is likely to change quite a bit in that we may not be required to make you submit a research question to us. It would take it out of the realm of research. It would treat it more like a regular agricultural crop. However, you would still have to come through us. We will still be regulated. In fact, if the law passes as written, we will have to submit our program to the federal government for their approval, and then we would be running it under the guidelines that are approved by the federal government. So there are still some changes to be made in the future, we think, with regard to how the program is run. Perhaps it will require less on your part with regard to coming up with a research program, but it will still be regulated. We will still likely have to come out and test to ensure that the THC levels are within the definition of industrial hemp. So having said all that, I know usually whenever I'm at one of these events, there's lots of questions that can take up lots of time, so I don't want to bore you with more stuff about the process, and I can just take questions now if you like.
There's a fixed rate of number of licenses for CBD right now. Is that supposed to be expanded or not?
Okay, the question is there a fixed rate for CBD licenses right now? There is not a fixed rate. We've just not been able to take, decision was made that we're not going to take any more for the time being. We expect that sometime in the future that we will begin taking CBD applications again. However, we're not sure when that will be.
How far in the future are you going?
We don't know.
So it could be years?
I don't think it would be years, but we don't know right now.
Do you know about regulations for CBD going across state lines? I know tincture forms are fine, but I think in terms of like food products, do you know the rules about that?
I do not know the rules, and this is in part, the question was, do I know the regulations with regard to shipping CBD over state lines in tincture form or other products like that? This is where some of the issue lies with regard to CBD production and the question of legality. And we always advise people that they need to get a good lawyer, a lawyer that works in cannabis issues. This environment is changing rapidly. The FDA recently approved a drug called Epidiolex for the treatment of seizures in epileptic children, and CBD is a major component of that, which now means that CBD perhaps will be looked at differently by the federal government. So we need to see what the federal government's going to do with regard to regulation of CBD.
Any insight as to the processing of grants? Is it closed down at random?
The processing grant program is run through Empire State Development. It's a different agency. I do know that they They did stop taking the applications for the time being. I'm not sure when or if that'll open up, but I think once again, it was because many of those applications were related to CBD, and we need to get a better handle on CBD.
Of the 3,600 acres, how many are actually planted?
We haven't yet got it would be out of 3,200 or so, and we're not yet sure how many were planted. We're in the process of having our horticulture inspectors go out to ground truth what actually was planted, and then, of course, they'll be back out at harvest time to take the samples that need to be tested at our lab.
Do you guys have any notion of how much is the space, the field space versus greenhouse space?
I don't know off the top of my head. The vast majority is field.
How is it that they sell CBD flowers right here in Geneva at 2 separate stores? How does that work?
That's an excellent question. The question was, how do they sell CBD flowers in a store in Geneva? That is not within our program, that's not within our purview, and that is a function of the lack of clarity with regard to the law covering CBD and other products.
And a lack of resources for enforcement.
More about how the state program is changed with the farm bill passes.
The question was, can I talk more about how the state program will change if the Farm Bill passes as it's written? It will require that we will submit our program to the federal government for their approval. It would also, it would take CBD, it would take industrial hemp out of the world of a controlled substance, which would make it less highly regulated on the federal side, but it would still require us to do sampling and testing to ensure that the THC levels were within the definition. And it would no longer, if it stays as it is, I believe, there would no longer be a requirement that it be a research project, and the restrictions with regard to commercialization would no longer be there. So right now, true commercialization of the product, according to the federal law, is not allowed.
But the state would still give you permits to grow and possibly.
That's my understanding. Yeah, we still have to have a handle on it because we would still have to know where everything is growing, and we need to sample and test it. So yeah.
Are the research licenses going to be renewable then at the end of 3 years?
The question is, are the research license renewable? Right. The research authorization is good for 3 years. It's a $500 permit fee, and then if your research project is not completed at the end of the 3 years, you can apply to us for an extension. We haven't yet had that happen because we haven't been at this for 3 years.
So with that $500 fee, that's going to stay the same if we get the 2018 farm bill moves through? Will there still be
The question is, would the $500 fee remain the same if the farm bill passes? I don't know the answer to that. I'm sorry.
The farm bill is not passed, is that what we're doing now?
If the question is, if the farm bill does not pass, would we cease what we're doing now? I don't think so. I think that we are pretty well on the road to being an industrial hemp state, and I believe that we would continue to work under the current program. That's a tough question. That would be my best guess.
Let's assume there would be a continuation of the existing farm bill.
If marijuana is legalized in your state, how would that?
The question is, if marijuana is legalized in the state, how would that affect? The programs would be two very distinct and discrete operations. There has been a work group formed by the governor's office to address the development of an adult use marijuana program. We're not sure where there may be overlap. There may be some in the CBD world, but we don't know yet. I think that work group is meeting today for the first time, actually, so we're very much in the formative stages there. The question is, are we still checking THC level if the crop is being grown for fiber and grams? Yes, we are, because the potential is there that it could go hot, and you would end up having.
it would be grown next year for the 2020 growing season.
Okay, but say that happens, they stay status quo, well, we're looking at, it's a height for all the we get overwhelmed by the whole process. So, but if we were able to buy it in New York State, would we still have to pick up all of them?
No, the question is if seed was purchased in New York State, would it still have to be picked up at our office in Albany? The only time that anyone needs to come to get their seed in Albany is if it's seed that's been imported from another country pursuant to our DEA import permit. And that's just the way.
Colorado, it can be shipped to us.
Exactly. At your own risk, because there is some gray area with regard to the federal law and various local enforcement authorities that you may meet along the way on your trip from Colorado to Long Island. There is some risk.
So when I mentioned that it is illegal to move across state lines, but that federal agencies could not expend money to intercept that, that does not preclude local law enforcement agencies from spending money to intercept it.
Anyone else? I've been ignoring this side. This is an area that the question is, if you're growing your CBD and you've done your extraction process, well, you've grown your CBD crop, it's sampled by our inspectors and tested by our lab, and it's within the 0.3%. But then through the extraction process, you come out with a product that is above the 0.3%. Is that then something that needs to be destroyed, or can it be diluted? That is an excellent question. And it is something that we are still trying to get a handle on, and it is part of the reason why we are no longer, at this point, taking the CBD application. Because, if it's above 0.3 THC, you're not within the parameters of the industrial hemp program. So,
New York, but Vermont, I think they are at 1%.
well, Vermont may be at 1%, but they might have gone rogue a little bit. We are held to the standard established by the federal law and the state law. And so there are other states that are taking a different approach, but, we're here and we have to do what we're told.
Do you have any numbers on what percentage as far as ratios for industrial hunters? Out of that 3,200 acres, how much is for CBD versus food versus fiber?
The question is, do I know how many acres out of the 3,200 are dedicated to CBD? I don't know the number of acres, but I know that out of the first 100 applications we got this growing season, 50 of them were CBD, so it was 50% at that time. We have been, taking food and fiber applications continuously, but it's a high percentage that are dedicated to CBD.
Just out of curiosity, there's a lot of interest in CBD, and some folks who are doing very large scale, and I'm curious if there is whether it's taxing or how the financial aspect is being regulated or at least observed, through a channel of the research, or is it just kind of, you guys watch the farm and after that the distribution?
Right. The question is there a taxation element to the growing or the producing of the CBD products or any of the industrial hemp? And no, there isn't. We do not have anything like that. And our agencies, we're strictly the ag side of things, but there are no special tax programs associated with the program.
A friend of mine in the medical marijuana industry in Colorado talked about that it makes a great deal of difference between the composition of the THC, CBD, based on the nutrient profile of the plant, the stress it's under, even the comparison of the same cultivar for change dramatically. Do you have any real intention how to keep CBD high?
Okay, so the question is about, from a grower perspective, can you optimize production of CBD while maintaining legal limits on THC? So that's an excellent question. Stress can induce that pathway. We don't have a lot of experience here at Cornell in studying that, but we do have 2 trials now underway to start to quantify CBD and THC production across a range of different cultivars. I'll be talking about that, and Chris Smart will be talking about that. And then also developmental stage is critical. Cross-stress can induce THC. So all of these things are critical questions to address. And all of these require quick turnaround testing labs, which we also do not have in New York State right now. So there is a regulatory testing that will be done in the food lab at Ag and Markets, but many of you growers want quick turnaround tests for THC in the middle of the season so you can judge when you should harvest so that you don't go hot. So there is, again, an urgent need for more testing labs, reliable testing labs in the state. I just have one more question for Tim, and then we're going to move on.
And I can hang out later.
And that question is, are you accepting applications for affiliated growers for CBD production from existing license holders?
No applications.
No affiliated growers either. So existing CBD license holders cannot expand.
Not at this time.
Right, just a quick question. Okay, I just want to thank Tim again. This has been an incredible partnership and collaboration with Ag and Markets and the legislature. Ag and Markets has never turned down a request that I know of to come and speak to a public group like this. They have been very open in working with us, wanting to develop a program based on the science, which I highly respect, and of course we're grateful for their research funding. So thanks, Tim. So a lot of those questions that folks were asking are very challenging in our current legal landscape. I serve on the Industrial Hemp Working Group, convened by the Commissioner of Agriculture. Mike Barnhart, who's here, also serves on that. I'm not sure there are other folks here on the working group, but when the governor accepted the report from the Commissioner on Health for proposing adult use marijuana here and the decriminalization of marijuana, that poses particular challenges for the industrial hemp industry, right? So if you're growing for CBD in particular, if your plants go hot, they're considered marijuana. So perhaps you will be better off if you're a CBD producer getting a marijuana grower's license. But we don't know what that program will look like, whether we will have outdoor marijuana production, and how that will interact with our existing medical marijuana program. So those are very challenging questions, and again, the state has been very prudent in rolling out these programs, and I believe that they're doing it again by sort of putting the brakes on the CBD production right now until we can work out the gold standard for regulation here in New York State. So we're going to move on. Jamie Crawford from Don Vine's program is going to talk about our cultivar trials. And then Matt Chartrand will talk about some of the pest surveys.
Okay, hi there. So this year we had 32 entries in our trials, while last year we had 17, so almost a double. That's pretty exciting. Hopefully it doesn't double again, because I don't know if we can really handle that many entries. But if any of you folks are growing varieties that you don't see on this list, please let us know, because I would be really interested to know if there are grain or fiber varieties in New York this year that we haven't been testing. It would be really interesting. So from these lists, you can see we got varieties from Canada, from Serbia, from the Ukraine, from Italy, from Poland, also some from Oregon. Things developed. There's some entries are pretty interesting. When the market started to expand, there was sort of a variety grab of European varieties. So United States companies have been buying up exclusive rights to a lot of European types. So one thing that we do have in our trials is Tigra. So we have a seed lot of Tigra that we purchased last year that we saved some seed from, and that was produced in Poland. And then we have Tigra that was produced in Kentucky last growing season. So we're going to have those two next to each other. And it'll be really interesting to see what the differences are, if any, just because of where the seed was grown. So we divided all of our entries up into 3 trials this year. So we have a fiber trial, that's pretty straightforward. But then we have a grain cultivar trial, and then one we're calling dual purpose. And those were divided by time to maturity. So based on the information given to us from the seed companies, anything that was 110 days to maturity or less went into the grain trial, and then more than 110 days went into the dual purpose trial. I'm so sad that you guys aren't able to see the variety trials, because I think that really highlights the importance of regional testing for the varieties, because we have 2 varieties in our grain trial that are not flowering yet. Everything else is about knee height and starting to set seed, and those 2 varieties are still not flowering. And then in our dual purpose trial, we have 3 or 4 varieties that are also starting to set seed. Well, the rest, and that was all just based on information given to us from the seed companies. So, yeah, so that just highlights how important the regional testing is. So this year we've got 6 different locations for our trials. So on the front you guys can see the map. We're growing some of the trial in Long Island this year, so we'll have some good information for that. We'll also have 2 locations here in Geneva, one in Ithaca, and then one on our organic free-build farm. So for all the organic growers, we hope to be able to provide some good information about that this year. And then the last trial is in Chazy, so just about as far north as you can get and still be in New York State. So that'll be some good information too. People that we've worked with in Canada have told us that varieties that are 8 feet tall in Canada are only 3 feet tall here, so maybe we'll get a little bit more height in our Chazy trial than we do here or in locations. So our first trial we planted last year was in, we planted it June 9th. June 9th was our first trial last year. And it was exceptional. It was an exceptional planting. This year, everything that we planted was within 2 weeks of that date. But the establishment wasn't, it was, I mean, you guys know, right? It was dry. We had a dry June and a dry early July here. And we went and we took notes, and just comparing this year's data to last year's, we had, it's 2 feet. In Ithaca, our trial is planted in the field directly next door to the one we planted last year, so everything should be the same. It was planted 4 days later, but you wouldn't think that would have that much of an impact. But the height, on average, the trial is 2 feet shorter this year than last year, and that's because we've had half the rain that we had last year. So in the first month, so the first 4 weeks after planting, we had less than 2 inches in Ithaca, and last year we had closer to 5 inches. So it was a really big difference. So I brought a plant. So this is a plant from our fiber trial last year, and it was actually taller. You can see the top of it broke off. We're not going to get fiber yields like that this year. It's just not going to happen because everything is so much shorter. I have I have confidence that our grain yields could still be very good. The plots filled in nicely. But our fiber, because of the drought, is going to be, right now the plants are about as tall as me. But I don't think, and they're starting to flower. So they're not going to get to be like this year. So one more thing I wanted to mention is the seed cost. So we're testing 32 varieties, and some of them cost just under $3 a pound when you include shipping fees, and some of them are over $30 a pound when you include shipping fees. So when we're just buying enough for our trials, that may not be a big deal, but I'm sure when you folks are planting many, many acres of it, that would add up really quickly. So I know there's people doing economic analysis of that as well, and we hope to provide that to you all as well. So you can see, like, maybe you get a little bit of a yield bump with something that you're paying $32 a pound for, but if it's not, if you're not getting the yield, you know, if it doesn't make up that initial seed cost, that would be very important to know. Yield, harvest is coming, so we don't have any information about that for this year, but it's coming soon.
But you might be a couple varieties that flower early on, so those also not very good. Flower later or flower later. That would be better for
So we are recording the maturity at different times throughout the year, so we should be able to report on flowering times for each of the different varieties. So what I was saying is that what we are noticing is differences in what's in our trials than what was in the company information. So we don't have that from one year to the next, but we do have that for this. We'll have that for this year.
And the flowering time, because we're covering almost 2 degrees latitude, the flowering time may be different in Long Island than it is in Jersey.
I'm just curious with the fiber, how much growth you saw after it started to flower last year. Was it significant, or
No.
did it basically stop?
It didn't stop, but so from that point on, it's just the flowering head that grows. So the part that would snap off easily and would not be really useful for the fiber reduction is what would still continue growing.
And we harvested pretty quickly after that.
So right after the
We don't want seed set. Within a week or two. Other questions?
Have you got any redding trials from the harvest last year?
Can you repeat the question? His question was, have we done any redding trials? We attempted to ret some of our material in Ithaca last year, and it's a learning process, I'll say. We did take stems out of each plot, and we made little humidity things in the greenhouse just out of plastic bags that we kept spraying to get it to ret that way. So we do have some of that information just on a few stems from each plot. But the field redding I think it's an art, and it's going to take some practice.
So this year for our fiber trials, we're working with Dr. Jared Nelson at SUNY New Paltz, together with Jen Gilbert-Jenkins at Morrisville State College. So together the three of us have defined redding protocols that we're going to use for our fiber trials. So we will field ret all of our fiber this year, determine a weight of straw, which we didn't do last year for each plot, and then share some of that material with Jared to do mechanical analysis on the fibers. And we'd be happy to share material with other folks to analyze as well.
Last year we gave all of our border rows to a gentleman who actually has his own pits, And so he water redded it, and he sent us some of the information that he received. But I think it was his first time trying it too, so it's very interesting.
Did you publish grain yield numbers from your trials last year?
I do believe that it is public. I'm not sure if it's on our website though, but we think it's on our website. He asked if we had published our grain yields from last year.
It's not in a peer-reviewed publication yet. Most of those agronomy journals require 2 years of data to publish it in a peer-reviewed journal. So I believe we're waiting to get this year's data, but
we do have tables, certainly of our graphs, so you can see the ranges of yields at the different locations based on different planting times.
Is that on your website?
if it's not, we'll get it
We'll
on.
make sure it's there.
Or I'd be happy to email it to you directly.
Last year there was huge variation depending on location. Like some locations were just like around 600 pounds per acre for the yield, so pretty low. Those were things that had poor establishment and then a lot of weed pressure.
Like heavy clay or ruby?
For this year or for last year?
Yeah, for both years.
Yeah, I mean, it's quite a range across different sites. And
We
drainage
do have
classes
that.
as well.
And the drainage classes and the soil types were definitely on the handouts from last year, and so those are on the website from the 2017 planting, and we can certainly add those to this year for you guys.
Thank you, Jamie. Jamie's the one who acquires all of our seed for us. That is a real challenge. Our plantings went in probably 10 days later than we would have liked. Again, because we're waiting for fiber seed to come from Italy. So that has been a real challenge. My number one piece of advice for all of you here is order your seed early. Try to get it in by February. Even from Canada, Keenan Stone is here from Uniseeds. The delays in getting seed from Canada into the U.S. seem to be even worse this year. So order your seed early. So Matt Chartrand from Elson Shields Group is going to talk about some of the sampling he's been doing for insect pests.
Hello, my name is Matt. As Larry said, I've been working with Dr. Elson Shields this summer. He's not here actually today, he has some other stuff going on, but I've been working to survey the insect pests that are out in the industrial hemp fields. So I think the best way to show you guys is this brochure here. So if you don't have one, there's a couple more up on the table up here to the left. So basically this whole summer what I've been doing is going out to various farms. I've been to 10 different counties so far this season to sweep for insects and kind of get an idea of what's actually out there in the fields and what may become an issue for growers in the zone. So on the center 3 panels here, you'll see there's 3 different insect pests that I highlighted. The first on the left is the tarnished plant bug, and that is by far the highest population pest we've seen in the field so far. And so there's a little bit of information there. I won't take too long to bore you with the feeding styles, but if you want to learn more about the tarnished plant bug, there's a little bit on there and an example of the feeding below. And then on the center panel here, we have the European corn borer. I've only seen this in one trial actually, and that was down in Orange County. And that was the most devastating insect I've seen so far. It only went after 2 plants, but it bored into the stem of the plant, resulting in death of the entire plant. And so that grower had to take out those 2 CBD varieties. So as you can see, and as Gary was talking about earlier, the disease in insects are going to be like a working document basically. So if the farmer sees something, take a picture or collect it, and feel free to send it along to me. And my email is on this brochure, or just collect it and send that actual physical sample to Cornell. So that's how we heard about the European corn borer. And then also we got the flea beetles, which seem to be in pretty high population as well. That's the far right panel on the center. And the flea beetles basically are vegetative feeders. They go after the leaves. You get those pinhole kind of feeding size that are in here. And so not necessarily something that's going to be harming your yield in the long run, but something to keep an eye on for you. And then on the center fold in here, we have the Japanese beetle. So if you're a grower around here, which many of you are out here, you know we have a lot of Japanese beetles in this area. And so the good news is I haven't seen it going after too many hemp plants, but that one example below is an example of what they can do. So it may be something to keep in mind for the future years of growing. If you see a high Japanese beetle population, traps or other ways to combat that breakout. And then one last thing I just wanted to mention, this whole project has been based on insect pests, so insects that are actually harming the plant. But I've been seeing a lot of natural predators as well, which is a good sign. So if we're seeing these high levels of pests out in the field, and it's also contributed to the high number of predators, that means that there actually may be a good sense of biocontrol in these hemp fields. So, as we all know, there's nothing listed for spraying on hemp as of right now, and it's going to be a continuing process of learning what's out there. So, we're at the mercy of your help. So, if you see something, please feel free to email it to us or let us know.
What proactive steps have you seen that have been helpful in containing or keeping pests at bay? Have you seen any things that have, just methods that have been helpful? Proactive is just sort of precautionary rather than, proactive rather than reactive on them.
Yeah. So the question is, what steps have been taken to assure that you're trying to help reduce these pest levels? And just overall good agricultural practices, controlling weeds, keeping weeds low, because a lot of that's going to attract other pests that are going to be attracted to your area. Natural rotation is a big one. So if you're growing in the same field over and over again, you're going to want to switch it out because then you're going to have the same populations of pests there as well.
I'm from the Cornell County area, and there's a lot of slugs and snails.
Okay.
How is that? How is that thrown into any of the larger growers?
Yeah. So the slugs and snails are a little interesting in themselves because it's not technically in the insect category. But I have been seeing them and spotting them when I do see them. One thing that I have seen from one of the farmers that I've been out to is diatomaceous earth. So it's a material that you can use basically as a barrier. When the slugs and the snails go over it, it tears them up. And so that's not necessarily a chemical-based control, but that is something that you can use culturally. Yes?
Were you aware of a predatory insect that you can use to these insects?
I'm sorry, did you say disease?
What eats these bugs?
Okay, yeah. So that's a big question. So these don't have a lot of natural predators, the ones that I've specifically highlighted here. The more of the things which makes sense why I'm not seeing them in high populations because of the natural predators, would be things like thrifts and aphids, more of the soft-bodied insects. So a lot of how these predators work is they'll inject their eggs into the soft bodies, and that will kill them from the inside as kind of like a parasitoid, and then also just feeding on the insects themselves. So the Japanese beetle, as many of you know, is pretty hardy, is a hard exoskeleton, so it doesn't have many natural predators. And so with these kind of things, I think the long-term goal is to figure out ways to control it, whether in terms of pesticides or other biocontrols that we can bring in.
Have you seen any more or less issues with greenhouse production?
So the greenhouse production is interesting. I've only seen 2 different trials that I've been to for greenhouse, and a lot of flies actually. The majority of flies are just attracted to the hemp plant itself. They're not necessarily causing damage as in feeding on the leaves, but they're a nuisance in the sense of trying to work to use bats and lots of flies in the greenhouse.
The fungus gnats.
Yeah, so the fungus gnats are actually, I haven't seen the high populations just because, the one greenhouse I was in was a seed house, or a transplant house basically, so they're partying them off to transplant outside. But the fungus gnats is something to keep in mind, especially with consistent water. You'll see a lot of fungus gnats.
Have you seen any spider mites in the field?
So I've only seen one case of spider mites, which is pretty good. But as you all know, we're kind of getting into the season where everything's flowering, and from the work done in Colorado and from the West, they see a high percentage of the spider mites coming after flowering, especially in the female flowers. So it's something to keep in mind as a research continues.
Thanks, Matt. So Matt is an intern funded by Cornell Crawford Extension and Cornell undergraduate. I just want to take this opportunity, because of Matt's ties with CCE, to mention that we have a number of our CCE hemp team specialists here, and maybe they could stand up. So Mike Hunter, Lindsay.
Katie's over there.
Kitty O'Neill, Mike Stanyard, Mary Ulrich, Janice Degny, who else am I missing? And John Hanchar. And John Hanchar, we made copies of John Hanchar's, Jody Laker. So it's been tremendous working with all these folks, and when you have problems, they can be your first line of defense. So by all means, get their cards and get their regional locations so they can help you out. Okay, so I'm going to talk about an experiment that we're taking on to try to address one of the important issues that faces hemp cultivation, especially as we move towards an adult use marijuana program, if it's going to be outdoors here in New York. Dave Williams at University of Kentucky, who is one of our primary academic researchers in hemp in the country, shared with me this unpublished data that highlights the key issue. That is, if you are growing for CBD production, there is a 2 to 3-fold decrease in CBD concentration in the flower shoots when the female flowers become pollinated. So that is your profit withering away when your female plants are pollinated. It seems to be even worse on some of the lower flowers than the top flower. The question is that the same with THC? Yes, essentially the entire pathway, and I'm not sure whether it's because of decrease in the production of the trichomes on those tissues or downregulation of the enzymes in the pathway. But the bottom line is you want to maintain, in a horticultural grow setting, you want to maintain your female plants unpollinated. That's a key question, I think, that we're facing. This is a key question for my breeding program, is whether we continue on in this horticultural setting in wide spacing from transplants, female plants planted with feminized seeds, which is incredibly expensive. So I had one grower tell me this is a $20,000 per acre investment in seeds and labor. Zach is shaking his head. So harvesting those plants by hand and drying them will add up to the cost, into the cost. The alternative is a more agricultural system in tight spacing, allowing those plants to get pollinated, perhaps capturing a grain yield in addition to a CBD yield, mechanically harvested, mechanically dried. That is the alternative. And I think we can breed for cultivars that produce even higher CBD. So last year, Futura 75, one of our dual-purpose grain and fiber cultivars, I'll call it a triple purpose, because it produced 2% CBD while also producing a reasonable grain. So I think that's an issue that fundamental cultivation issue that growers will address. We hope to breed cultivars that can make your choice a little bit easier.
Industrial hemp is wind pollinated. Nate will come up and talk about some of the bees that he's seen in field so far this year. But those bees that are attracted to male flowers are not attracted to the female flowers. There's no nectar production. The pollen is very light and dry, so it's carried by the wind. In the Midwest, where there is a lot of ditchweed, feral hemp growing around in Iowa, Illinois, Nebraska, Minnesota, Wisconsin, hemp pollen can be more than 30% of the pollen that's trapped in late summer. So it is a major issue. Fortunately, we do not have very much feral hemp in New York State. We collected from one feral population in Fort Wayne. I've heard about a couple of others that I will investigate here in the next month, but we have very little feral hemp. So that is not a major concern in New York State. The major concern would be if your neighbor across the street plants a grain and fiber field of a monoecious or dioecious variety that's going to produce a lot of pollen, and you're directly downwind with your CBD or in the future outdoor marijuana. The question is, how far will that pollen move? So there are anecdotal stories of pollen blowing from Morocco to Spain. It's a critical issue for certified seed production also. So we can look to the AOSCA standards and the standards in Canada for certified seed production. I believe the AOSCA standards are a 3-mile isolation zone in Canada, 5 kilometers. There is no academic research on this topic, very little. So there's one paper by Ernie Small at the University of Toronto, where they took glass slides coated with grease and trapped pollen at various distances from hemp plants. And that graph is in the handout. You can see that over 10 meters, there is only 20% of the pollen ring compared to next to the plant. At 400 meters, there is less than 1% of the pollen that is produced at the plant. What they did not do is test whether there is sufficient pollen to pollinate those female plants. Because even less than 1% of the total pollen produced may be enough to get full seed set. So what we have done here, this is a challenging experiment because of the size of our farm. We planted a 3-acre field of Anka over here in the rain, and then planted female plants of 3 different cultivars, proposed to be high CBD cultivars, in microplots, one of them right next to the Anka field, so it should get pollinated. And then in the 4 ordinal directions at 200 meters away and at 400 to 500 meters away to the northwest and south. On the east side, we could only go 200 meters to our property line, so we jumped to 1,500 meters to the east to our next available planting spot. So we will monitor those plants for signs of pollination. At the end of the season, we will quantify seed yield by plant as a measure of functional pollination. And we will also extract cannabinoids and quantify CBD production by plant. So this will give us sort of a dual-purpose experiment. We will get a lot of site information, genotype by environment information on CBD production, because we've got 9 different sites here. And we will also be able to verify Dave Williams' data about the 2 to 3-fold drop in CBD production upon pollination. It's our first attempt. I think, the conditions here in New York State, with our topography, with our moist climate, so to speak, compared to Oregon and Colorado, where they talk about 10-mile pollen movement, I think our distances will be much shorter than the potential distances in Oregon and Colorado, but we need replicated data to validate. I'll
stop there and I'd be happy to take any questions.
If you have a grow and someone applies for another application for a particular fiber or food, will that application be turned out?
So the question is, who is going to manage the geographic arrangement of fiber plantings, grain and fiber plantings, versus CBD and potentially marijuana plantings? The state ag and markets has already said that they will not regulate that. They are not interested in imposing regulated pollen exclusion zones to the preference of CBD producers over our grain and fiber producers. What I would suggest is at meetings like this, you get to know your neighbors, because in my opinion, the best way to regulate that is neighbor to neighbor. But sometimes that's not going to work, and you may need to work it out through other means. That may be talking about rotating fields, varying distances, compensating someone not to plant in one field in preference for another field. The ultimate assurance is to move your plants indoors.
But according to the hoop house, in here it's green, there's something that's planted up out.
Sorry?
If you're doing hoop house production, if you're insect screen or some kind of screen that you can put on the hoop house to keep pollen out.
Yeah, so the question is, how much risk are you for pollination in a hoop house, for example? And there's absolutely no data on that, but
Is there a product out there that has planted up the screen?
hemp pollen is very small. I think you have to have a very small mesh, but are you aware?
I know a guy. So yeah, in terms of hoop house production, or really screens to keep out pollen, I know a bit about this, that people are looking for screens to keep out pathogens. And depending on the pathogen, the pollen and the pathogen may be of similar sizes. So I know one vegetable grower who wants to keep all pathogen spores out of his high tunnels, hoop houses, and he actually put HEPA filters on the ends of the high tunnels, and that has been extremely effective. But, so he grows tomatoes. And tomatoes are really happy at 120 degrees Fahrenheit. So if you're trying to grow hemp for CBD at 120 degrees Fahrenheit, chances are your THC levels are going to go through the roof. So and I think that HEPA filters are the only filter that's going to be effective in keeping pollen out.
Other questions? Yeah, so I think the question is about its isolation distances for certified seed production. So, Ian, maybe can you talk about how Canada regulates isolation zones?
So for certified seed production in Canada for industrial hemp, we actually have 3 levels of isolation distances. The closest for just certified seed production is 1 kilometer of the same variety. If it's a monoecious cultivar, it has to be 5 kilometers if it's isolation between a monoecious cultivar and a dioecious cultivar. It has to be 5 kilometers isolation if it's different cultivars. If it's the same cultivar but of a higher pedigree, it can be, I think it's 3 kilometers isolation between those different pedigrees. So you start from breeder, so you have breeder, foundation, registered, certified in terms of the level of pedigree.
The inspection for seed certification in New York State. Other questions? Nate, are you ready? Nate or Heather?
Heather in the back.
Yep.
trial spot is described. You can measure THC before pollination possibly, and then after pollination.
So the question is, will we measure THC before pollination and after pollination? So we have not collected any samples yet, and there is active pollen production in our field. So our intention is, our hope, is that some of these microplots will not be pollinated by the end of the season. And those will be our measure of non-pollinated plants.
We discussed how CBD is reduced after the plant is pollinated. I'm wondering, is that also a way to possibly save the crop that the THC has gone too high? When you pollinate it, you might have less THC, so you save your crop without having to destroy it because you THC.
Oh, so the question is, could you recover from a field that's gone off by pollinating it? I don't know where you would, at the end of the season, where you would all of a sudden get all this pollen. But it's a good idea on a small scale. If you had plants elsewhere that were actively making pollen. One more question about pollination. One more question about pollination.
There's no data from Colorado or Oregon, California?
There are no published peer-reviewed papers.
But just like talking to growers?
Yeah, again, you talk to growers and they'll say, you've got to be 10 miles away from me. So it's anecdotal. I have not, so I've only been doing this a year and a half. I have not encountered anyone who has told me, yeah, my field got pollinated and ruined. I just haven't encountered that yet. So there's a lot of hype. I don't know how real it is. What I have encountered is folks from Europe who have said, I have my field here, my neighbor has his field over there, you can see it. They are never cross-pollinated. Okay, it's my pleasure to introduce Dr. Heather Graff, intern, and they're going to talk about pollination.
Thanks, Larry. Can everybody hear me okay? Wonderful. All right, so given that Larry just told you all that hemp does not need pollinators, and in fact probably suffers from having pollinators, you might be wondering why I'm up here telling you about pollinators. Probably, actually by show of hands, I would love to see how many of you have heard about pollinator concerns, saving the bees, all that good stuff. Awesome. So that's what we're working on in our agroecology lab down in Ithaca. We work on all kinds of beneficial insects. But we've heard quite a bit about all of the bees that are showing up in these industrial fields and trials. So we decided to come out and take a look and see what's going on. Not because we think that having bees in hemp fields is a benefit to farmers per se, but we think it could actually be a huge benefit to the bees that are out there. So a lot of research has been going on, figuring out why we're experiencing so many kinds of bees, has identified lack of pollen, lack of floral resources as a major key that are out there every day pollinating your crops, but they're also looking for other things to eat. When strawberries aren't blooming, apples aren't blooming, squash isn't blooming. And we know that during the window when hemp is starting to flower, that's a period of time in New York State where we have very few floral resources on the landscape. So potentially as we grow this industry, we're starting to add a huge resource for our wild bees back into the landscape. So part of what Pete and I are doing this summer, as things are starting to bloom, is to go out and survey in fields and figure out which bees are benefiting. And it may not be that yourselves are benefiting very much from having those bees there, but if you grow other crops, if your neighbors are growing pollinator-dependent crops, if you have an apple orchard that's blooming in the spring, strawberries, raspberries, squash, field tomatoes, any of these other crops, the same bees that we're seeing right now in the hemp field are the bees that are the most effective. So by supporting them in a period of time when there maybe isn't very many other goals around, we could actually be contributing to supporting their population overall and have a long-term beneficial effect, not just for wild bees in general, but also for crop production outside of hemp. So I'm going to hand it over to Nate, and he's going to tell you a little bit about the details of the project and the survey that we're doing right now.
Yeah, so the main kind of motivation behind the project is with this new huge surge of hemp in New York State. How is it essentially going to be affecting the bee communities we have here right now? And pretty much you've seen so far that hemp, or bees really do love hemp. Like Larry was saying, hemp is completely mispollinated. Hemp doesn't have extra floral nectaries, so bees can't collect nectar from it. They're only getting pollen. We haven't seen bees visit female flowers, so the bees are not pollinating the plants at all. But we have been seeing them go to hemp in mass. Like we've seen a lot of bees there, bumblebees, honeybees, and also a lot of native bees. So what we've done is we have 10 farms around Ithaca, Geneva, and Seneca. And we're going to be visiting them 3 to 4 times with beesmaster bees. We're going to be collecting all the bees we catch, bring them back to the lab, and identify them as species. And then we're going to look at them in these crop networks, like Heather was saying, and see essentially where the bees that we see at each site overlap with other crops in that landscape. So yeah, we're also hopefully going to be looking at floral resources within the landscape to see essentially how much bees have in that site and whether that's influencing them coming to those sites. Yeah.
The handout has a couple photos of what it looks like. They're really collecting huge amounts of pollen from those male plants, and a list of some of the species that we've seen so far. But hopefully as this project progresses, you'll hear more from us next year about what are the crops that would be most beneficial to actually have them growing nearby in order to get that benefit of a boost in pollination in those kinds of crops. So, great question. The question is about what happens when bees, particularly honeybees, which are the ones that are producing all the things that we like to eat, are visiting these crops, collecting pollen, bringing it back to the hive where it may mix in with honey. And so far we have no data on what that means in terms of honey quality. There's certainly lots of things that we are tracking otherwise that are coming in, so we're worried a lot about pesticides that bees are picking up, which fortunately, given the current regulatory status of hemp, we don't have to worry about them bringing back fungicides, pesticides, and other things back in the lab. But are they bringing in COMP-PE or PhD in pollen? We don't know, but it's certainly something we should look at.
But there's so much ditchweed in the Midwest.
Yeah.
There must be a lot of
question in the back?
We have 72 hives on our property right next to the hemp field. So we've really seen maybe 2 or 3 bees on hemp plants. They're all avoiding it, basically. So I don't know if any of your sites actually have bees right next to the hemp field or not.
So we're hearing about a scenario where we have a lot of hives right next to a field of hemp.
9 acres.
9 acres. Okay, and that's something that we have been noticing is when we're on really large fields, like even the field that we had down the road here, 3 acres, we see many fewer bees per plant in those kinds of scenarios. And it's probably not that they're avoiding a field so much, it's that they're just diluted in the field. So we don't see them as often as we would when we're looking at a small trial that's just a micro plot planting where there's really a ton of bees. But it's also
probably a factor. Maybe your farm has something else that they like better nearby. Honeybees are social. They can recruit foragers into really high quality sort of areas. So they might have something better right now that they're going to, but that's not always the case. All right, thanks.
All right, we are in the home stretch, gang. So if anyone does not have a handout, Larry can give you one. It looks like all the others, more or less, but it says evaluation of industrial hemp cultivars for CBD. And I was really looking forward to having folks up at our CBD trial this morning because, you know, when I started this a couple years ago, I heard, oh, hemp can grow anywhere, hemp grows fast, hemp. We got some plants that are downright dead. And I think it's important for you guys to understand that, you know, there are places where hemp loves to grow, and there are places that hemp is really unhappy. And so where we have one of our CBD variety trials, if you look at the back of the sheet, you can see the list of varieties. The varieties that were planted were started in the greenhouse because we wanted to ensure that we had germination and plant establishment. And actually Larry clapped at that, the folks on the stairs over there, Team Hemp. And the plants were then in storage pots and planted into the field, and the dates were early July. July 6th in our Integrative trial, and July 9th in Geneva. So when those were planted, we were just at the end of the very dry period. So we had, they're on raised beds with drip irrigation and plastic, and we were irrigating them, and then it started to rain. And one end of the field that they're in is just a wee bit lower than the other end. So the water drained that way. And the plants that had wet feet for an extended period of time, even though they were on raised beds, some of them flat out died. Some of them are extremely stunted. And as you go down the field, when you get that slight incline, the plants at the end that was just a bit drier are doing quite well. So I think it's really important to know that that concept that you can plant it and it will grow is not always the case. So that's an interesting trial to see. The reason that we're doing the trial is to answer some of the questions that many of you have asked in terms of, you know, what impacts CBD levels and what impacts THC levels here in the state of New York. If you're growing for CBD, you don't want your plants to hit above that 0.3% THC level. And you know, we have information from Colorado and Oregon and Kentucky, but we don't have information on the stressors in New York. For example, the plants that have now had wet feet for much of their lives in the field, are they going to have a higher THC or CBD content because of that flood stress? And we don't know. To compare, we actually are growing the ultra-rich CBD, the 7 ultra-rich varieties from Nevada. We're growing those in our greenhouse. And what we want to do is compare the yield as well as the CBD and the THC levels of the greenhouse-grown plants to those plants that were grown in the field. And for this year, we'll compare the Ithaca trial, the Geneva trial, and the greenhouse plants. And I saw some pictures that Jamie, I think, showed that the plants in the field, you know, plant architecture is one of the things that we're looking at in these varieties. So in a heavy rain and wind down in the Ithaca trial, was that last week, Jamie? Some of the plants actually snapped. You know, the stem was just not strong enough to handle the foliage and the plant architecture that was above. Oh, that would be dramatic. So what we're measuring on all of these plants right now, we are measuring establishment. If they die, we're measuring plant architecture, and we're doing that by counting the number of nodes where leaves would come out on the plant and then the number of nodes that have branches. So that gives you an idea of how branchy the plant is. For some people, they're looking for a big branchy plant that looks more like a Christmas tree. And for other folks, they want maybe a single head. And so we want to be able to provide you with, in New York, in our state language, what is the plant going to look like? And that's really the goal of this study. So we'll have plant architecture, we'll have height, and in the greenhouse, what are our tallest plants now? 2 meters?
Something like that, yeah.
Yeah, and in the field, they're roughly the same. So we have plant height as well as architecture, and then what we'll have is not only CBD levels, but we hope to have the whole panel of cannabidiol, cannabinoid compounds, so that we can give you an idea of what is being produced under the conditions, each of the conditions that we had our trials in. As we move forward, we hope to then be able to compare. Next year, we hope to have some plants in high tunnels. Right now, our high tunnels are in a place that gets rather wet, so that will have to be modified. But it would be great to be able to give you guys information on high tunnel production versus greenhouse production versus field production, and that's what we really hope to be able to do with some of the varieties. And then as new cultivars come along, new genetics in the plant, we hope to be able to test, as Larry said, one of the things we really want to be able to do with experiments like this is to test rapidly. So we can, sort of on the fly, understand how stress conditions are modifying the secondary metabolites, or CBDs, in the plants. So one of the things that my lab is doing along those same lines is we're looking at the hemp microbiome. So you've heard of the human microbiome. We all have microbes on our skin and in our stomach, and that affects many, many attributes of our health. The same exact thing is true in plants. And one of my students, a PhD student, Ali Kalla, has looked last year at the microbiome of the hemp plant. And she has now, we think, identified sort of the microbes that are on hemp. And she looked at 6 different locations, and we have sort of a core microbiome. And that's sort of a starting place in terms of understanding what bacteria and fungi are on plants. And these aren't necessarily pests. They're just present and interacting with the plant. It's known from other systems that the microbes on the plant can modify things like drought tolerance, flowering time, and secondary metabolism, which could modify our THC or CBD levels. So we really want to know what's there. Additionally, if there are beneficial microbes that are being, that are associating with the roots of plants, if we can identify some of those, then we can work with Al Taylor, who is an expert at using things like that to actually enhance plant growth. So those are some of the things that we're working on. One thing that Ali found that I thought was absolutely fascinating was that in the microbes that are found on the flowers of the hemp plant, the most common microbes are those microbes that have previously been reported from extreme environments, like hot springs and high salt. And so there's something, the secondary metabolites in the flower of the plant that the microbes that associate there are those that can handle extreme environments. And so that's something that we'll be looking into as well. So were there other things about the variety trials?
And flowering time also.
Oh, right, for sure, flowering time. We're noting that both in the greenhouse and in the field, and the field plants have flowered earlier than the greenhouse plants.
Was it all seeded or during long?
Sorry?
Was it all seeded plants or during long planting?
We do have one clone, but it wasn't planted after the field. Everything was from seed.
How many of the varieties are autoflower versus photoperiod?
So there are some autoflower. I don't know how many.
I think we have, 4 of them are autoflowering.
When the data comes out, will that be...
Yeah, that'll be included, for sure. But we have cultivars or lines that still have not flowered. So we have both males and females in the population, and as soon as we see a male, we cut that out. And there are cultivars that have not flowered yet. We don't even know if the plants are male or female, August 15th. In the back.
In the future, will you try varieties that come from places that plants have wet feet?
Great question. In the future, will we try varieties or cultivars that can handle more rain? And if we can find those, absolutely. But I think that's going to be one of the things that our breeding program that is led by Dr. Smart, will look at, along with flowering time and daylight.
Artificial lighting in the greenhouse?
Yes, so we do have artificial lighting in the greenhouse, but it is not longer than the daylight. Did you want to add?
Just using nitronal from what you're managing.
Right. So that's one of the things that we hope to get out of our high tunnel experiments next year is, can we manage moisture via high tunnel, and how does that compare to greenhouse production? Yeah, so the question is, did the dry period at the beginning of the year cause excess stress on the plants, causing them to flower earlier compared to those in the greenhouse? And I think it's a combination of the dry at the beginning followed by prolonged wetness that added the stress. Yeah.
You talked a lot about your harvesting plan. People participating in the program have to be noticed 20 days in advance, so how we can plan better for that.
Our harvesting plan, right. So how do we decide when to harvest, and how do you decide when it's 20 days before our harvest so you can let Tim know? We've had a lot of discussions on how we're going to do that, and how are we going to do that? How are we going to, when it looks like they're going to get stressed, and certainly we need to harvest before frost. And so that will be taken into account. And my guess is that we'll probably, depending on the weather, say sometime between mid to end of September, and then bank our 20 days on that. Yes?
You were talking about the plant structure, the height. How do you take into account cooling and popping and
Right, great question. So I was talking about plant architecture and height, and how do we take into account any pruning that might happen? And so we are not doing any pruning at all. There are, of course, some leaves that drop, but we're not doing any pruning on this for this particular trial. Yeah. So in future trials, in future trials, we'll be doing pruning to make clones. And for my part of the experiment, I won't be doing pruning to make clones. I don't know, Larry, do you have thoughts on that?
So the issue is the material transfer agreements under which we obtain this material. So if we are restricted against cultivating it and propagating it from cuttings, we won't do that. For that material that we are allowed to make cuttings, we will probably try to do that and again use that material for breeding as well.
All right, thank you very much.
I just want to mention one more project that we're working on, which is to, I'm sure many of you have seen the Phylos galaxy. If you haven't, you should go take a look at it. Phylos Biosciences, that's with a P-H-Y-L-O-S, is a company based in Portland that will analyze the DNA of your hemp or marijuana plant and compare it with everything else that they've got in their database. And I think they have thousands of samples in their database. So it's very cool to look at which cultivars are related to one another genetically. It's like a 23andMe or Ancestry.com analysis for cannabis. Phylos, P-H-Y-L-O-S. They have a very cool web interface where you can sort of zoom through the Phylos galaxy. So we are trying to do the same thing for the CBD cultivars that folks are growing in New York State. I have already collected small leaf samples from, I think, 4 different farms to extract DNA, to do that similar type of genetic fingerprinting analysis. And you see we're already up to 35 different named cultivars. The question is, are those true to type? None of these were purchased under a certified seed program that would verify that they are true to type. How related are they to one another? How distinct, is there genetic differences between these that would be of interest for us in our breeding program? So I know there are a number of growers here I want to connect with where I haven't been able to sample yet. If they're willing to have me go and collect some small leaf samples for DNA extraction, that is something we hope to be able to publish next year. Is when you buy Cascade or Lifter or Electra from the Crawford brothers, is it really any different from buying cherry wine or cherry blossom from Centennial Seeds? Off the top of my head. Okay, I think that brings the formal presentations to a conclusion. Again, I'm really sorry we can't make it out to the field, but I would like to open it up and give folks an opportunity. If you need to take off, you can take off, but if you have questions, I'd like to open it up for a general question and answer session now.
This has been a production of Cornell University, on the web at cornell.edu.